President Min-Chi Hong, distinguished speakers, honored guests, ladies and gentlemen, welcome to the 2024 Tang Prize Master Forum in Biopharmaceutical Science. The topic of today is contemporary development of type 2 diabetes treatment and obesity control. We are honored to have the 2024 Tang Prize laureates, Professor Svetlana Mojsov and Professor Jan-Jules Holst, to be here with us. Before we start today's program, we will have a group photo first. Please remain in your seats, and our photographer will take a photo from the stage. Everyone, please turn to the stage, look at the camera, and smile. Thank you very much. In the beginning, I would like to invite academician Hong Ming-Qi, the president of China Medical University, to deliver the opening remarks and introduce our distinguished speakers. Ladies and gentlemen, let's welcome President Hong.
I'm not sure I should wear glasses or not. And for those of you who are senior, you know what I'm talking about. When you come to this age, sometimes you need the glasses, sometimes you don't need the glasses. I'll try not to use it. Okay. Dear Tang Prize laureate, Professor Svetlana Mojsov and Professor Jan-Jules Holst. Of course, the laureate's family, and also the Tang Prize Foundation, the CEO, Chen Zhenjuan Jiao, and also the convener, Tang Prize Biopharmaceutical Science academician, Zhang Wenchang. All honored guests, and my dear China Medical University colleague, ladies and gentlemen. It is a great pleasure and honor to welcome all of you to join this 2024 Tang Prize Forum on the contemporary development of the type 2 diabetes treatment and obesity control in Taichung. This year, the Tang Prize in Pharmaceutical Science was awarded to Professor Joe Habener, Svetlana Mojsov, and Jan-Jules Holst, three of them, for their seminal contribution to the development of type 2 diabetes treatment and obesity control, namely famous GLP-1. Dr. Joe Habener from Mass General Hospital couldn't visit Taiwan this time, but his original cloning of the precursor of glucagon gene from anglerfish in the early 80s has initiated a multiple decades of research and finally finalized in the GLP-1 truncated peptide 7-37 amino acid, which later on you will hear from the laureate. And that particular peptide was shown to be effective to treat obesity and diabetes patients. Currently in the world, there are 500 million diabetes patients and almost 1 billion obesity patients, including myself. Okay. And Professor Mojsov, she's a world-renowned peptide biochemistry from Yugoslavia. She received her PhD degree from Rockefeller University in 1978 under supervision of the late Robert Bruce Merrifield, who received a Nobel Prize in 1984 because he's developed solid phase peptide synthesis. And Dr. Mary Field actually visited Taiwan in 1977-78. Professor Mojsov continued her postdoc work with Mary Field in Rockefeller till 1983. Then he moved to Boston, joined the Mass General Hospital from 1983 to 1990, where as an independent researcher, she runs a Howard Hughes Medical Institute peptide core in Mass General Hospital as an independent researcher. And it was there that she started her very active and productive collaboration with Professor Habener and many others to search active GLP-1 peptide that can stimulate insulin release. And through her ingenious expertise in peptide biochemistry, she unveiled the mystery of GLP-1, namely a truncated form of GLP-1 to remove the first 6 amino acid, so-called GLP-1 7-37, is the one that associated with the activity to stimulate insulin release. Therefore, opened a serious collaborative work to develop GLP-1 into an active treatment for type 2 diabetes. Starting from 1990, she was back to Rockefeller University to serve as an assistant professor. In 2002, she was promoted to be the associate professor. And lately, due to the recognition of GLP-1, which is so important for humankind, her contribution has gradually and correctly been recognized and received many, many prestigious awards recently, including this Tang Prize, and a few weeks ago in US, Lasker Award. Trust Rockefeller University will soon, well, she just told me that Rockefeller already did a lot of things for her. But I think, personally, I think that Rockefeller should very soon give her an endowed professor title, which she deserved for a long time. Of course, to search for an active intestine secreted peptide hormone to stimulate insulin release for treatment of the diabetes patient, this is not only in the US. It's an international event. For example, in Europe, in Denmark, Professor Jens Juel Holst in the University of Copenhagen, his team was also actively pursued on this direction. His lab would show GLP-1, the peptide, in pig intestine has the activity to stimulate insulin release, and also show that this peptide affect human appetite. So you will not, even you go to night market, you will not like to eat. Okay, so you can control your diet. Okay, so his lab is still very actively working on this area, on GLP receptor agonist, one direction. The other direction is the DDP4 inhibitor, which I believe he's going to present it, so I will not mention here. Professor Holst received his doctorate degree for medical science from University of Copenhagen, also in 1978. But he was an assistant professor then. Then in 10 years, in 1989, he quickly ranked to the full professor. And through his entire career, he has been very, very productive. I'm a little bit jealous, but anyway, I'll later on say it. That, he has been well recognized by his peers and have received many, many awards. So in 1993, from his country, from Denmark, okay? In 1993, he was a member of Danish Academy of Natural Science. 1996, he became professor, but that year he became member of Royal Danish Academy of Science, and in his country, this is the highest honor. In 2010, in University of Copenhagen, he served as a senior group leader, Novo Nordisk Foundation Center for Basic Metabolic Research. And when we talk about Novo Nordisk in Taiwan, it's equivalent to Taijidian semiconductor. So that company in Denmark has made so much money to become a magic mountain to protect the country. And in Taiwan, there's another one. It's what the Taijidian is called what? MSTC, yeah. That is also a magic mountain to protect Taiwan. So he was a senior group leader over there. And as I mentioned, he has been very productive in his career. And I was a little bit, actually not a little bit, I was very jealous because his number of publications is more than, listen, 1,900. And his Google Scholar Edge Index exceeds 200. Okay. With this very brief introduction, I would like to send my wholehearted congratulations to both the laureate, Professor Svelina Mojsov and the Professor James Joel Holst, for their well-deserved Tang Prize Award. And I would also like to call, by the way, congratulations. And I would like to call the entire audience to join me to give them a big applause. Thank you. Thank you.
President Hong, thank you very much. Please take your seat. And now, let us watch a short video about the 2024 Tang Prize laureates in biopharmaceutical science to learn more about their contributions.
In the early 20th century, the concept of incretins was taking shape. Scientists realized the intestines may secrete hormones that cause insulin release and regulate blood sugar levels, but their nature and functions remained a mystery. In the early 1980s, Professor Joel Habener at Harvard Medical School, when cloning the pre-pro-glucagon gene from anglerfish, found it contained another peptide sequence.
Which was very much of a mystery and a surprise, and it was not glucagon, but it was very close to glucagon. We called it glucagon-related peptide.
Later, he cloned this highly conserved gene from mammals and discovered glucagon-like peptide 1, GLP-1. Its role was identified by Professor Svetlana Mojsov, head of peptide synthesis core facility at Mass General Hospital. When she saw the 37 amino acid sequence of GLP-1, she saw the 6th as a possible cleavage site.
If one is to cleave that arginine residues, there will be a shorter peptide of 30 amino acids, which I called 7 to 37. And I predicted that that's going to be the active sequence.
She later proved her hypothesis and synthesized GLP-1 7 to 37. In 1986 and 87, Mojsov and Habener published 2 major papers demonstrating that truncated GLP-1 7 to 37 is bioactive in inducing insulin release and isn't an incretin. At about the same time, Professor Jens Joel Holst at the University of Copenhagen also identified GLP-1. Holst found that after GI surgery, patients' insulin rose while blood sugar dropped, attributing it to incretins.
We purified the real peptide from the body of humans, and purified it and put it on the pancreas, and it worked. And it turned out to be the same peptide, of course.
The 3 scientists together confirmed that GLP-1 7 to 37 had great potential for diabetes treatment, but the problem was GLP-1's short half-life. After more collaboration in R&D, a GLP-1 receptor agonist was developed. Such agonists were also found to aid in weight loss, sparking a revolution in treating diabetes and obesity.
Addressing the glucose but you're also modulating weight loss, energy utilization, appetite, gastric emptying, and a number of different things. So for the first time, we have something that has multiple effects that modify the disease process.
Since GLP-1 receptors are present in several organs, the drugs may be applicable to cardiovascular diseases and cancer as well. The future of GLP-1-based therapeutics is bright. The biomedical contributions of these three scientists are truly far-reaching.
Next, I'd like to invite 10th Prize laureate Professor Svetlana Mojsov to deliver a talk entitled Chemistry Matters, from a putative peptide to efficient therapeutics for diabetes and obesity. Ladies and gentlemen, let's give a warm welcome to Professor Mojsov.
Well, this is a tremendous honor for me to receive the Tang Prize for my contribution to the discovery of GLP-1 and to, I have to start this, okay? Okay. So, and to actually have this opportunity to show you my experiments. Do you hear me? Yeah. This opportunity to show you my experiments which led to that discovery. So my interest in how peptides regulate glucose metabolism goes back to when I was a graduate student in the 1970s with Professor Bruce Merrifield at the Rockefeller University. We were actually interested in understanding the role of glucagon in glucose metabolism and diabetes. Parallel experiments in the field were focused on the discovery of incretins. So what are the incretins? Incretins are peptides which were secreted in the intestines in response to a meal or high glucose load taken orally, and then which secrete the insulin release from the pancreatic beta cells and thereby influence glucose metabolism. Now, in Merrifield's laboratory, we were interested in understanding more the glucagon biology. And what is glucagon? Glucagon is a peptide of 29 amino acids and is secreted in the pancreatic alpha cell and stimulates glucose output from the liver and thereby increases the circulating glucose in the blood. So our approach to understand glucose, the biology of glucagon, was to synthesize by chemical means large amounts of agonists and inhibitors. And the solid phase peptide synthesis developed by Bruce Merrifield in the 1960s was the method of choice. So in this slide, I will show you briefly the principle of the peptide, solid phase peptide synthesis and how I applied it to the synthesis of glucagon. So the idea is to attach the last amino acid to a resin support, and that's threonine. And it's insoluble, and then to attach every subsequent amino acid one at a time until the entire sequence is assembled. And then by the number of chemical reagents, we cleave the peptide from the resin and purified by chromatography. So my PhD thesis was to develop a novel and very efficient synthetic strategy which will allow us to obtain very pure glucagon in high yields. Subsequently, I developed a second improved strategy to which was even better in obtaining high yields of glucagon. And by the time I completed these studies, I knew the glucagon sequence backwards and forwards. And it was really this knowledge of glucagon sequence and glucagon biology that was critical for my discovery of the active sequence of GLP-1, which I will show you in the next several experiments. Now, other investigators in the field were using a molecular biology approach to study glucagon biology. And in the summer of 1983, Graham Bell published the sequence of the human glucagon gene. And what he showed that glucagon is made as a part, that glucagon is made as a part, glucagon is made as a part of a larger molecule, which contained, in addition to glucagon, the sequences of 2 unknown peptides at the time. And it was Graham Bell which named them glucagon-like peptide 1 and glucagon-like peptide 2, or GLP-1 or GLP-2. And you can see in this blue panel, Bell predicted that GLP-1 is a peptide of 37 amino acids. And other investigators in the field came to the same conclusion, including let me show here, Gerhard Heinrich in Joel Habener's lab when they published the sequence of the rat GLP-1. Now even, this is not, can somebody help me here? Oh, yeah, 2, okay. So even in 1986, Orskov and Holst and their colleagues reported that GLP-1 is a sequence of 36 amino acids. However, when I reviewed the sequence of the GLP-1, I noticed immediately, I'll show you better here, the presence of an arginine residue, which is here, in the predicted sequence of GLP-1. And I predicted that the cleavage of the arginine residue at the beginning of the sequence will lead to a shorter peptide of 31 amino acids, which starts at the position 7 of the predicted sequence. And that's shown much clearer on the next sequence. So here I will explain in more detail the rationale. So this is the sequence of 1 to 37. And if you can compare it to the sequence of glucagon, you can see that there is very little sequence homology between 2 of them. But if it's going to be cleaved at this arginine position 6, we are going to get 7 to 37. And when we align it with glucagon, we will see that there is a perfect alignment between amino acids in 7 to 37 with those of glucagon, which were known by that time are important for the biological activity of glucagon. In particular, I knew from my work at the Rockefeller University the importance of the histidine at the beginning of the sequence of glucagon. If that histidine is removed, then glucagon becomes inactive. And then everything came together for me. I predicted that GLP-1 7 to 37 would be the biologically active sequence, and that will be the incretin. So what is an incretin? It should be secreted by the intestine. It should stimulate insulin release by the pancreas, and the insulin release should be glucose dependent. So the first question we ask, well, is 7 to 37 secreted from the intestine? And to do that, I developed a strategy to prove my hypothesis. So this is outlined the steps which I undertook. So the first step was to obtain by the solid phase synthesis large amount of 7 to 37, and to use this synthetic material obtained from a single synthesis for all the subsequent studies. So that involved developing methods to detect it in the rat intestines, that was the model system, and also to use it in characterization of this biological activity. And for that, And as I will show you later, we used the perfused rat pancreas. And we showed this, I will show you in great detail these experiments. I'll show you the results, because that were the experiments we showed that 7 to 37 is an incretin. I also collaborated at that time, started a collaboration with Joel Habener's lab and his postdoctoral fellows, Drs. Dan Drucker and Jacques Philippe, to look at the expression of GLP-1 and its biological activities in cell lines. However, those experiments were inconclusive, and I will not show them to you. And then finally, and then finally, let me show here, this is moving fast. We actually used a GLP-1, the synthetic one, in clinical studies. So let's start. So the first step was to obtain to synthesize 7 to 37. And for that, I used the manual shaker. Now, in 1983, manual shakers were not available commercially. And Dr. Merrifield sent me one from his laboratory. The next step was then to use the synthetic peptide to develop very sensitive radioimmunoassay, which is really a quantitative measurement of the concentration of 7 to 37 in tissues and in the blood. The next step was then to develop chromatographic methods to separate the larger 1 to 37 from the shorter form, 7 to 37, and from all the other peptides which are present in the gut. And for that, I used first gel filtration chromatography, which separates by molecular weight. As you can see here, I actually detected them in the intestines, but the gel filtration chromatography is not sensitive enough to separate them by molecular weight, because if you can see here, they're different only by 6 amino acids. So the next, however, they're different in charge. So I already mentioned the arginine residue. There is an aspartic acid. There are 2 glutamic acids. And to separate them by charge, I actually developed something which was new at that time, high-pressure ion exchange liquid chromatography. And as you can see on the rat panel, not only that I separate them, but I actually detected 7 to 37 in the intestinal extracts. So that solved the first question. It is secreted by the intestine. The next question was then, but will it stimulate insulin release? And for that, I started a very productive collaboration with Dr. Gordon Weir from the Joslin Diabetes Center in Boston. And when we injected very small amounts, 10 to the minus 11 molar, very low, the same one which exists in the blood, we actually saw a sharp increase in insulin. And this is much more obvious when we go to 100-fold higher concentration. You see, now we can measure the amount of 7 to 37. As that concentration goes up, so does, look, the black line, the concentration of insulin. And in the same experiment, we also use the longer 1 to 37, the predicted form. And you can see it's flat. We are going to 10 to the minus 9 molar concentration. This is the concentration now of 1 to 37, which we can measure by radioimmunoassay, no insulin response, and even remarkably at very high concentrations which don't exist physiologically, there was no insulin release. So that solved the second question. It stimulates insulin release specifically by the pancreas. Now, in subsequent experiments with Gordon Weir, we showed the stimulation of insulin release is dependent on the glucose concentration. As the glucose concentration goes up from 6.6 millimolar to higher 16.7 millimolar, we see increase in insulin release. Notice the difference in the units on these 2 different axes. And this is really what you expect in a real situation. because when you eat a meal, you have increase in insulin release in order to compensate for to control the circulating glucose levels. And another important point from this experiment is that at very low concentration of glucose, it's inactive. And this was very important in the terms of thinking about it, because it meant that if a medicine would be developed based on the 7 to 37 sequence, then it's not going to be active at low glucose concentrations. It will prevent hypoglycemia, which is typically observed with insulin treatment. And with this experiment, we showed that 7 to 37 is an incretin. It's secreted by the intestine, it stimulates insulin release by the pancreas, and the release is glucose dependent. Now, what was really important conclusion for this set of experiments, that is actually the glucagon gene, which includes the incretin. And Jens Holst in Copenhagen was interested in the same problem, worked at the same time, and he reported that another form of GLP-1, 7 to 36 amide, stimulates insulin release from the perfused pig pancreas fourfold. Notice at 10 to the minus 9 molar concentration. Now, at that concentration, Gordon Weir, in collaboration at 10 to the minus 9, he also saw that actually glucagon also stimulates insulin release fourfold. Notice here the different units on the y-axis. And in contrast, at the same concentration, GLP-1 7 to 37 stimulated 20-fold. And so, and of course, that was even more pronounced when we went to this very low concentration of 10 to the minus 11 molar. And what this experiment showed, that 7 to 37 was the most potent peptide, insulin-releasing peptide detected at that time. Now, I was interested in understanding, let me go here. I was interested actually understanding, we have to go one back, in understanding the biology of 7 to 36 amide. Because 7 to 37 and 7 to 36 are different only by this one amino acid, in which arginine at position 36 is being amidated. And then we went, and in my laboratory at the endocrine unit, we synthesized 7 to 36 amide, and using the same methods which I applied for the synthesis of 7 to 37, I showed that it's actually present, 7 to 36 amide, both in the rat intestine and to a smaller degree in the pancreas. And in subsequent experiments with Gordon Weir, we actually showed that they're equally potent. Here we again on this very, very low concentration of 10 to the minus 11 molar, and we showed that equally potent, very high insulotropic effects. Now, many years later, Mojsov and Holst showed that in human blood, both forms circulate, and that 70% of circulating GLP-1 is in the form of 7 to 36 amide, and 30% in the form of 7 to 37. Now, at the same time, Gerhard Heinrich, working independently in Joel Habener's lab, showed that identical messenger RNAs are present in the pancreas and in the colon. But of course, the Northern blot analysis is not sensitive enough to detect which peptides are expressed in which tissues. You needed the analytical methods, which I developed, to show that in the intestine, we have the three forms of GLP-1, the two active ones, 7 to 37 and 7 to 36 amide, and the second glucagon-like peptide, or GLP-2. While in the pancreas, the main product is glucagon, and to a smaller degree, the three forms of GLP-1. So, let me go here. So clinical studies after that followed very quickly. And by the end of 1987, Kramer and his colleagues in Stephen Bloom's lab showed that 7-36 amide is an incretin in men. But we were interested at the Mass General to understand, wait, you're going ahead of me. This is not showing up. We were interested actually to find out where we are now. This is Kramer's experiment. So we were interested, it somehow doesn't show here, but we were interested to find out whether 7-37 has any effects in type 2 diabetes. And for that, we collaborated with Dr. David Nathans from the Massachusetts General Hospital. And this was the first clinical study. It was very small, but it showed that the infusion of 7-37 led to a high increase in insulin and decrease in blood glucose levels. And so our experiments were quickly confirmed, first by the group of Suad Efendic and his Scandinavian collaborators, and later on a very important study by Michael Nock in Creutzfeldt lab. So, but my interest was then to understand whether 7-37 we have biological activity beyond the pancreas. And what we showed with my colleague Yang Wei, that identical GLP-1 receptors are expressed in the pancreas, in the brain, in the heart, and the kidney. And this showed for us that if a medicine is going to be developed based on the sequence of 7-37, that is going to have other effects beyond the pancreas. But there was a problem with 7-37. It had a very short half-life, and it was quickly degraded by an enzyme DPP4, or dipeptyl peptidase 4, to remove the first two amino acids and to lead to a compound which is inactive. Now, if you remember, I showed you before that histidine in glucagon was very important for its biological activity. And it showed here, again, the importance of my prediction, that it has to start from position 7. Once it's degraded, then it's not active anymore. So this was solved first actually by the group in pharmaceutical companies who developed small molecule inhibitors of the enzyme. And the first oral drug which came to the market was sold now, sitagliptin or Januvia, it was developed by Merck. And it's a daily pill which is taken, which is even still in use. And what was very important, this was actually the first GLP-1-based therapeutic because it prevented the hypoglycemia, which was typically observed with insulin. Now, Novo Nordisk solved this problem and extended the half-life of 7-37 first by 13 hours, and then remarkably, this was really a remarkable accomplishment by the scientists at Novo Nordisk, to 1 week. And they did that by attaching a long chain of lipids, and which then led to the first medicine, liraglutide, which was taken once daily, and then to semaglutide, which we all know now is Ozempic, and it's very efficient in use for weight loss. So now, in 2024, we have a whole range of GLP-1 analogs, which are used to treat diabetes and obesity. The one is blue, is from the Novo Nordisk, and the later newcomer is the one which I want to point out, and maybe we can bring during the discussion, is this nirseptide, which is developed by Eli Lilly and based on the work of Richard DiMarchi and Matthias Job. It's a second generation of GLP-1 agonist, but it's a combination of 7 to 37 and GIP structure. So it's one molecule which can treat these two different conditions. So in conclusion then, GLP-1 agonists are a first example in which a single peptide can be used, or a single medicine in this case, peptide-based medicine, can be used to treat multiple human disorders. From diabetes, we know now for the suppression of appetite, weight loss. Even there are some studies which show that it can treat Parkinson's disease. And of course, we know that there are very beneficial effects on the cardiovascular effects and effects on the kidney disease. And so my expectation now is that this is really presents the beginning in which we can see development of other peptide-based therapeutics for treatment of human disorders. Now, finally, I just want to mention my acknowledgement slide. First of all, as already mentioned, I'm very grateful to the Rockefeller University, and I want to acknowledge my mentors, the late Dr. Bruce Merrifield and the late Dr. Ralph Steinman. I also want to thank the Howard Hughes Medical Institute and the late Dr. George Cahill, who really supported all my work at the Massachusetts General Hospital in the 1980s, which led to the discovery of GLP-1. Now, I also need to thank my collaborators. And I didn't have a chance to show you that work, on the studies of better understanding the biology of GLP-1 and glucagon. Also, in the Massachusetts General Hospital, my deep gratitude goes to Henry Kronberg, the head of the endocrine unit, and Dr. Henry Koitman. And especially, I want to thank this group of my young, talented, and very dedicated research assistants who worked with me side by side on the bench during the initial stages of GLP-1. And finally, deep gratitude to Gordon Weir and the members of his group, whom I work on studies to understand better the biology of GLP-1. Again, studies, a lot of them which I didn't have time to show you. And this is the funding which I've received throughout the years. So thank you very, very much.
Professor Mojsov, thank you very much for the stimulating and inspiring speech. Let's thank Professor Svetlana Mojsov again. Next, I would like to invite 10 Prize laureates Jens Jules Holst to deliver his talk entitled Incretin Hormones as a Basis for Treatment of Obesity. Ladies and gentlemen, let's welcome Professor Holst.
Thank you very much for this invitation. Today, rather than repeating myself from the Tang lecture, which was about the past, I will concentrate on what happened during the last 25 years with the GLP-1 research. So let's get on with it. So this is the incretin effect, right? That's the amplification of insulin secretion, which is seen when glucose is taken in orally as compared to intravenously. We all know that. And what are the incretin hormones? So we have GIP on one hand, and we have GLP-1 on the other hand, and they are very similar in terms of insulin secretion and beta cell function and beta cell even proliferation. But if it comes to glucagon, they're different. One stimulates, the other one inhibits. Also, the GI effects are different. And in type 2 diabetes, we have pronounced differences. So this is interesting. So how to find out what they do? And the way to do this, really the good way to do this, is to use peptide antagonists, receptor antagonists. So this was possible, and here we have exendin 9 to 39, which were by brave researchers who dared to inject this peptide from the poisonous venom of the Gila monster into people in 1999. I'm really impressed about that. They did this, and that can be used to block the GLP-1 receptor. And we were fortunate enough to be able to identify and isolate a GIP receptor antagonist so that we could look at both of the receptor system at the same time. So here we have the GLP-1 receptor agonist, the Gila monster, as I said. It's a full and very potent receptor antagonist, and you can see here that it activates the receptor. It's not the GLP-1 of the Gila monster. It has its own, and nobody really knows why this peptide is produced in the Gila monster. And you can see here that it's a very nice antagonist. Here you see the antagonism produced by it and the binding curves over here. So this is the GIP receptor antagonist that we developed in Mette Rosenkilde's laboratory. She's sitting right here in the front row. And here you can see that it's a potent inhibitor and that it has no agonist activity and that it has a competitive action looking at this hill plot. That's how you do that in technical terms. So this is what why it's so useful. And now comes the critical experiment. So now we're looking at people. And now we're looking at a glucose tolerance. And we can then block one receptor at a time, you know, the GLP-1 receptor, the GIP receptor, and both of them, and see what happens. So here we have the control experiment, and then we block one receptor, and that impairs glucose tolerance, and the other receptor also impairs glucose tolerance. And if we block both of them, you will have glucose intolerance. So these incretin hormones are really important for glucose tolerance. They're essential for that. So which one is the more important? And if you look at the insulin responses in these experiments, you can precisely calculate which one was more important. And if you take away the GIP action, it's responsible for 45% of the insulin release. And GLP-1 is not quite as important as the GIP, only about 30%. And the glucose takes care of the rest. So the problem here is the DPP-4 inhibitors, when we are trying to translate this into the DPP-4 here, you have it. Sorry, so we just go on back here. So we have here the DPP-4 that cuts away these two amino acids. You already saw that. And we found that, and we demonstrated that in 1995. And we demonstrated that it has a half-life in people of 1 to 2 minutes, which is ridiculous. It has a clearance rate of 3 times the cardiac output, which is also ridiculous. I mean, but nothing else is eliminated that quickly. And the kidneys are there to eliminate GLP-1 also. Alone that will give them a half-life of 4 to 5 minutes. So what can we do about that? How can we use this for clinical medicine? And this is what's been looked at. So over here we have some peptides that are stabilized against DPP-4 because they have a different amino acid here, and are also stabilized against the very extensive extraction in the kidneys, but they still are filtered in the kidneys, and that means that they have a half-life of 30 minutes in the circulation. So something had to be done, and that's what's done in various ways. So over here we have the Novo Nordisk approach, that is the acylation. You already saw that. And if you improve the acylation technology, you can come up with semaglutide and a week's half-life, which is really, I was there developing semaglutide, and it was extremely exciting. So dulaglutide was from Lilly, and they put a big protein, an FC fragment here, attached it to 2 molecules of it, and that, of course, prevents the filtration in the kidneys and gives it a long half-life. Tirzepatide is, again, an acylated peptide here, but it's a combination of GIP and GLP-1, which is interesting, and maybe we can come back to that. So here is tirzepatide, and I'm using this as a very, very good GLP-1 agonist, and that's what it is. So come, have a look here. So in these people with diabetes that are treated with tirzepatide, half of them with a high dose will get an A1C level, a glycated hemoglobin level of less than 5.7%. That is normal. And that means that you can treat diabetes back to remission with a GLP-1 agonist if it's sufficiently good. And also in these diabetic individuals, you also had a great weight loss, as you can see here. So how does this fit into the treatment of type 2 diabetes? And that's what I really like to present here. So how do you treat diabetes? You treat them with lifestyle modification, diet and exercise. There's metformin, there's sulfonylureas. They're about to die out now because many drugs are better. There's the incretin-based therapies. There's the SGLT2 inhibitors, very important. There's insulin, of course. And then there's bariatric surgery. I was voting with some other colleagues which one was best, and we said, oh, bariatric surgery is best, but that's another story. So do they really help? Let's have a look at diet and exercise. And if you look at that, here's the most famous study of all that. That's the DPP program. And you can see here over a 10-year period what happens with diet and exercise. Yes, there is a very nice reduction in the mortality rate here and here over this. And this is metformin in the middle here. So it works, not tremendously, but it does work. So lifestyle modifications really do help. Well, this was then tried in the big look-ahead trial, which is one of the most extensive and expensive trials that has been done in this. So several thousand people were looked at, 5,000, with diet and exercise for a long time, for a 10-year period. Everything was done to get optimal diet and exercise and lifestyle therapy. And as you can see here, weight loss was pronounced in the beginning, but it was difficult to maintain. At the end, after 10 years here, we ended up with some 4 or 5% weight loss. So what happened? What happened? So there was nice effects on body weight, and as you saw, fitness function, glucose control, quality of life. But the major important outcome, the major cardiovascular adverse events were not changed. After 10 years, there was absolutely no effect of the therapy. So lifestyle changes didn't work. So this was the situation. This is one of the most depressing results ever reported. If you look at it, and the trial was stopped because of that. If you look at those without cardiovascular disease at the baseline, these were diabetic individuals, there was a p-value of 0.06. And if you look at the data, you can see, yeah, it's creeping to the left side of this forest diagram up here, but it's not significant. It's the confidence limits are crossing the one line, so it really doesn't work. Those with cardiovascular disease at the baseline, they were even worse, of course. So it didn't work. But then it turned out, thank you. And then it turned out that after 10 years, if you looked at those that had had a 10% weight loss, then they would have a 21% reduction in the cardiovascular complications, the end part. So what it meant is that if you have a sufficiently large weight loss, it will help. But if it's not sufficiently large, it would not help. So this was, of course, extremely important. Here's another extremely important result. That is the direct trial from Scotland, Roy Taylor's study, where he looked at a dieting approach in people with type 2 diabetes and made these individuals diet for a year, and they saw a remission of diabetes in a quite large proportion. And it's even more dramatically shown here, because here you have the weight loss from the dieting, 5%, 10%, and up to 15% here. And those that maintained a 15% weight loss had an 85% remission rate of diabetes. So 85% of the diabetes cases went away if you could have a weight loss of 15%. So that tells you how important it is to have a weight loss of this magnitude. I'm sorry. So doesn't A1C, doesn't glucose regulation matter at all? Yes, it does. But you can tell here from studies with insulin here and sulfonylureas that if you have increasing A1C levels, you will also have an increased risk of dying. So it's really, really important, A1C. But apparently weight is even more important. And here, if you look at time in range of blood glucose and complication rates, you can see that the less time you have in range in the regulated area, the higher the risk is of getting complications here, retinopathy, and here microalbuminuria, that is kidney disease, of course. So everybody would try then to improve glucose control, and therefore big trials were started. Here you have them. The UK PDS, the ProActive, the Advanced, the VADT, and the Accord trial, in total 33,000 people that were treated with optimal therapy to lower their glucose levels. And here you see the A1C levels going down nicely in all studies. A very nice all-over study from 7.5 to 6.6, a strong effect on A1C. And did it help? Let's have a look. Here we have the forest plot. And here you can see, no, not significant at all. If you look at the overall result, absolutely no effect on cardiovascular risk and mortality. On the contrary, the Accord study, there was an increase in the mortality rate. People started to die because of the therapy. Okay, that was bad, of course. Then came the SGLT2 inhibitors, the glucose reabsorption and the kidney inhibitors. And they work, of course, by inhibiting the SGLT2 here in the kidneys. And as you can see, sorry, let's have a look at it again. Here you can see that here we have the SGLT2 sitting. And if you can inhibit that, you will also inhibit you can increase glucose excretion with the urine. And here you have the result of that study, the cardiovascular outcome trial with the SGLT2 inhibitors. That is a sensation. It worked. It saved lives. It prevented cardiovascular deaths. It prevented heart failure. And the overall major adverse cardiovascular effects was reduced by 14%. So they presented this study in 2015 in Stockholm at the EASD meeting. And it was a sensation. We were all completely crazy about it. We were standing on our chairs and waving because this was the first time it was possible to treat the mortality and the complications of type 2 diabetes. So what then happened, of course, was that the GLP-1 receptor agonists also were introduced. And 1 year after, we had semaglutide and liraglutide showing a very nice reduction in the risk of MACE, of cardiovascular problems. So here we have the 26% reduction with semaglutide. And this is perhaps the most important slide I'm going to show you at all. It's really, really, really impressive. So if you look at the meta-analyses that have been made about this, so the short-acting ones, they don't work really, so forget that. But all the other ones, they will have a nice effect on cardiovascular outcomes here. And if you look at the details of these, it's MACE, the major, myocardial infarction, stroke, cardiovascular mortality, all-cause mortality, and even heart failure, and all of them are very significantly improved. So this is the promise of the GLP-1s. Particularly stroke is interesting, because this is something that you cannot do a lot about, where you have the SGLT2 inhibitors over here, nothing with stroke. DPP-4 inhibitors here, nothing with stroke, but the GLP-1 receptor agonist, a very nice and significant effect. And here you have a summary of semaglutide effects in stroke. You can see the very pronounced effects of semaglutide on the risk of stroke in people with diabetes. So here is another extremely interesting study. That is the risk of developing dementia while you're on therapy. And see how strongly it is reduced during these many, many months, 130 months of therapy, a huge study from Sweden with 50,000 individuals. So this is what you can expect. Kidneys is one of the biggest problems in diabetes. That's the kidney disease. And over here you have the trials with semaglutide here and the reduction in the risk of developing kidney disease, as you can see during these therapies. Very, very important. And here is the FLOW trial, very recent trial of semaglutide. Thousands of people looked at over several years. And you can see here, very importantly, we're looking at glomerular filtration rate, which is kidney function the way you should study it. And you can see that in the controls, you have a decrease in glomerular filtration rate. That's the impairment of kidney function in people with diabetes. But you can certainly inhibit that process with semaglutide, as demonstrated there. So what about heart failure? And these are the results of the SELECT trial. Again, sensational, because the GLP-1 receptors agonists, they have an overall effect on the risk of heart failure, which is very much reduced, as you can see here. But it also shows that it is both the preserved ejection fraction, which is typical of type 2 diabetes, and the reduced ejection fraction of heart failure, which is very much improved, as you can see here in both of these graphs here, with the GLP-1 receptor with semaglutide. So how does it do that? How does it do that? That is, of course, the extremely interesting question. And we've already talked about some of the effects of the GLP-1 receptor agonists. And we've talked about the weight and the glucose regulation, and the blood pressure is also nicely affected. But I'd like you to focus here on inflammation, which may be one of the most important parts. You know, you have inflammation in obesity, you have inflammation in type 2 diabetes, and all the inflammatory markers can be reduced during the therapy with the GLP-1 receptor agonist. And also you have the vessels, the blood vessels where there are receptors for the GLP-1, and they are apparently involved in the health of the endothelial cells, and they will prevent the atherosclerosis progression when they're excited. So that's probably some of the most important effects of the GLP-1 receptor agonist, to interfere with inflammation and to improve cardiovascular function. And that explains that you can have all these enormous effects on different organs, the brain, the heart, the kidney, and the liver. So What about the body weight, which is, of course, also, as I already showed you, a completely essential effect here? So let's have a look. This is a dose-response study with semaglutide. Because, you know, semaglutide had a nice effect on body weight, but what about if you increase the dose a little bit? Let's see what happens. So this was done in this experiment. This is the placebo, and here comes Saxenda, the 3 milligram liraglutide dose, very nice weight loss, as you can see. But then comes increasing doses of semaglutide, and now we have responses of up to 14–15% of body weight. And that inspired people to start the STEP trials with the 2.4 milligram semaglutide dosing. And you can see here the results of the STEP 4 trial here, where there's a very nice effect of almost 18% body weight loss. And remember, the 15% was the one that was required to have an 85% remission of type 2 diabetes. So we are there with that kind of therapy. And this was then studied in people with obesity, but no diabetes in this huge SELECT trial where 17,600 people were followed for 40 months, and where the risk of type 2 diabetes was decreased by 73%, but importantly, the risk of cardiovascular disease was reduced by 20%. So even in people without diabetes, you will have a great improvement of the risk of cardiovascular disease and mortality. So this is what it looks like in the SELECT trial. This is the weight loss. And have a look, there's a very nice weight loss here. And it's maintained for 4 years. There's no tendency of return. So it really, really works. Of course, tirzepatide is also very, very good for this. And here is a very similar study, the SURMOUNT 4 trial, where we are looking at the effects on body weight here for 88 weeks here and a 26% reduction in body weight. Really a huge result. So what is the future for these agents? Well, it takes up to 2 years to reach the target in terms of body weight. And some will have a slow onset of action, but be patient and work with it because it will work. It will work. So it's essential in this period where you up-titrate the dosing to change lifestyle. That is when you should start exercising, because otherwise you will experience a big loss of lean body mass. But you can counteract that by changing your lifestyle to more exercise. And you can do it because you lose weight and it will be easier. You also need to change your diet. And again, the good news is you can do it because you have lost this murmur of the food, this demand to eat all the time. It's gone. So now you can change it. You can start eating cabbage. So what to do after the weight loss? And again, that is extremely important. If you have instituted lifestyle changes with exercise and diet, then there's a better risk that you can maintain the body weight loss. If you don't do that, you cannot maintain the body weight loss, that's for sure. So that's one of the approaches to do this. So who should have it? And here I'd like to emphasize that the patient that should have these medications is this guy. He has the metabolic syndrome. This is a patient that comes in, with central obesity, elevated triglycerides, low HDL, BP, but blood pressure slightly elevated, plasma glucose slightly elevated. He has the metabolic syndrome, and this is about 25% of all grown-ups in Europe at least. The risk of heart disease, stroke, and diabetes in these people is increased by 2 to 3-fold. And if you have all of these, your risk of getting diabetes is increased by 25-fold. Is there an effective treatment? Yes, there is. The GLP-1 receptor agonists. So what I've told you is that there are 2 incretin hormones, GLP-1 and GIP. The best of the compounds we have today may cause diabetes remission. In obese individuals, they will result in weight losses of 20 to 25%. Weight losses of this magnitude may cause diabetes remission, which is the main important message here. The GLP-1 receptor agonists reduce cardiovascular risk and offer renal and neuroprotection. The adverse effects are usually mild, also long-term. So what do we do now? What do we do now? What are we going to do with this? So there's a huge job ahead of us to try to find out what we should do for the society. And this is, of course, a political, cultural issue, an economical issue. How do we solve this problem? How do we provide this medication to all these 25% of the grown-up? That is a job for all of us to discuss this. So thank you very much for your attention.
Professor Holst, thank you very much for your amazing and inspiring talk. Now it's time to move on to the panel discussion and question and answer sessions. During this forum, our moderator will be accepting questions through the Slido. Please scan the QR code or click on the link and below the live stream channel. Thank you very much for your cooperation. Ladies and gentlemen, I would like to invite to the stage the host of this forum, Academician Hong Min-Qi, the president of China Medical University, to moderate the panel discussion. I would also like to invite all our panelists, the 2024 Tang Prize laureates in biopharmaceutical science, Professor Svelena Mojsov and Professor Jens Jules Holst, Academician Shitu Huikang, president of National Health Research Institute, Professor Zhuang Limin, distinguished part-time attending physician, Department of Endocrinology and Metabolism, National Taiwan University Hospital, and Dr. Wendy Huang, CEO and President of the Board of Anji Pharmaceutical Company Limited.
Nobody tell me that I have to start it, so I can start myself. Being a university president is not unusual, or is quick. Nobody tell me what to do. I usually have to wait for somebody to tell me what to do. But anyway, we just heard the two great talks for this very important discovery, and apparently for human disease, it's still continue ongoing. And thanks to the two laureates. And today we have an excellent panelists whom I will just briefly introduce from my right-hand side. Zhuang Limin he's from National Taiwan University. He was a physician and having treated a lot of diabetes patients. And also, very importantly, when we were little, we were neighboring. So we know each other when we were in elementary school. And then from the other end, Wendy Huang, who is the CEO and the president of the panel from Anji Pharmaceutical company. So she's the person who responds for making money. So interesting making money, talk to her. And then the last person, but not least, is the president of the NHRI, National Health Research Institute. He's also academician of Academia Sinica, Shitu Huikang. Thanks for joining us. So we may have 40 minutes or so. So what I would like to do is, by the way, thank you for the two great talk. So maybe if we start from that end, then we go to a couple rounds. Then after that, if possible, I would like to save 10 minutes for audience to ask the question. And for audience, you have to use Slido, okay? You will see that. Did anybody use that QR code, so Slido? So if you have questions, prepare to do that. So why not let me start it first, okay? So Wendy, you want to go first?
Everybody can hear me, right? So I was supposed to be the last, Hong Xiaozang, but I'm so honored. I'm very happy I get to ask first. And after listening to the two lectures, I hope two laureates doesn't mind I modify my question a little bit than I previously talked to you, and the same question go to both of you. Being a pharmaceutical product developers, I'm particularly interested about where we go, like Professor Holst said at the end. So now it's being developed as an oral drug, as a dual active, as a combination with a GIP agonist, with a GIP antagonist. So where are we going as far as the next superstar of this field? That's question number one, okay? And question number two is, it sounds like a panacea to all. Will we see it on a vitamin shelf a few years from now? I think everybody should take a pill of GLP-1. So with that, please.
So I think we already have, which I mentioned with the TIRS peptide, we see that a combination of two peptides is actually better in reducing weight than the single one. So I can see in the, so that's a combination, so just to, again, review, it's a single molecule which combines the sequences of 7 to 37 and the second incretin peptide GIP. And we can see they act on two different receptors. So we have a single peptide which does that, and it has a higher efficiency in reducing body weight. And as I mentioned, I actually think there will be other peptide-based therapeutics would be explored, and we already know that pharmaceutical companies are working on that. Another, we should consider really bringing leptin back to the picture, because we know that leptin is very efficient in reducing weight in healthy subjects, but not in patients with type 2 diabetes. But if, say, semaglutide or tirzepatide can bring the weight down to what can be considered a healthy state, then leptin can maybe continue to actually act on different tissues through the adipose and also combination between the brain and the adipose to actually maintain that weight. And so I see that there will be a combination not only of different pancreatic and intestinal peptides, what we are testing, what we are looking now, but actually including other proteins or other hormones which can influence actually weight.
Okay, so that's an interesting proposition, and I'd like to add that one of the actions of amylin, which is now one of the components that are being combined with GLP-1, is reported at least to resynthesize the sensitivity to leptin. And that may be one of the reasons why amylin is a particularly interesting combination product. But you're right that there are many new developments underway. And for instance, there's GIP agonist and there is GIP antagonist, which is really hugely interesting. And right now, it is not clear in people why this is. There's a lot of studies in rodents, and it's absolutely clear that rodents are different from humans. They are, really. And that makes it very difficult, actually, to find out exactly what goes on. Now, if you take tirzepatide, the effective dose is 15 milligrams, and they compare it with 1 milligram of semaglutide. So you say, what's the difference? It's the difference of 15 times the dose. Nobody thinks of that. I mean, that's ridiculous. So that Novo Nordisk company right now, they're trying higher doses of semaglutide to see if they can obtain the same. And we don't know that yet. That's very, very possible. The actions of GIP are very, very controversial, and we don't really know. Right now, so what happens with people with genetic variations in the GIP receptor is that all the inactivating receptor mutations are associated with leanness. And this is completely consistent with the old concept that GIP was the obesity hormone. So this is not a logical component. What happens with tirzepatide, it's a wonderful drug. It's really, really potent. It is a super GLP-1 agonist. That's what it is. And we can be happy about that because GIP story is, it's different. So that's one of the interesting things. The other one is, will it be in the supermarket? Can we just buy them? And that's a very good question. So one thing is that there will be many, many, many sites where these drugs will be produced in the future because there are factories all over the globe that are ready to produce it. And in addition, there will be small molecules, most likely. There's a small molecule in phase 3 of orforglipron, which is very, very promising. And it's much easier to produce the small molecules in large quantities. So that will be accessible in a completely different scale. But you're right, should it be in the supermarket? Well, I don't think so, because the way I see these medications, they are to prevent complications of diabetes. And that means that you should see your doctor and get a prescription if you are at increased risk of this. And that's how they should be used, in my mind.
So you think the food additive is probably not an option right now? Okay.
But but come on. Once you have developed visceral obesity, you're up the risk ladder, right? So you should control your own metabolism very carefully and see if this begins to affect your blood pressure, if it affects your lipids and your blood, and then you may be eligible for GLP-1 therapy.
Okay, thank you.
To add one more thing, producing semaglutide or liraglutide with lipids, it's a very complex procedure. The yields are very low because the lipids tend to stick to purification methods. So I think what we need to think now is to develop new chemistry in which we can extend the half-life of 7 to 37. To just think in that way, and I'm sure, the efforts were based on the lipids, but I think we need to start thinking in terms of chemistry back. And I think that will come along. Something which is cheaper to produce, something which is easier to purify. And I think one of the why there are shortages of semaglutide is because it's very difficult to produce it on a scale for millions of people. And so I think there will be combination there.
Thank you both. Very inspiring for chemists.
Okay, thank you. So maybe next, academician Situ.
Yeah, I'm Hui Kang Situ from National Health Research Institute, Taiwan. So again, thanks a lot for the excellent presentation, both of you. So providing us actually very, very comprehensive information and also the innovative conceptual framework about the seminal discovery about the pro-glucagon gene, including the GLP-1, and also the subsequent identification of the biological function for the truncated 7 to 37 peptide as the insulinotropic peptide. So I'm curious, based on the whole discovery process, there are two interesting phenomena. So one is that, as you mentioned, the 7 to 37 GLP-1 peptide actually, based on the amino acid sequence, it's similar to glucagon. So, but they actually displayed totally different function. So the glucagon to increase the glucose level, but the truncated GLP-1 actually lower the glucose through the insulin stimulating property. Moreover, these two functionally opposite molecules actually put in the same RNA transcript. So I don't know if you have any other idea to further elaborate for this kind of mechanism in terms of the evolutionary advantage or regulatory advantage for this kind of processing mechanism. Yeah.
So both actually glucagon and GLP-1 are two hormonal peptides which actually connect our nutrients with glucose metabolism. So when you eat a food, when you eat a meal or take a lot of glucose, like take a sugarcane drink, you need a quick response for the body to lower glucose. And that's what GLP-1 does. It's secreted in the intestine and stimulates insulin release and quickly reduces the glucose levels. Glucagon, on the other hand, I think is a hormone which acts on the liver. And when you don't consume food, I think at the times when you didn't have constant access to food, but there were periods of starvation. And even in modern days, we can see that various religions require fasting during the day. Glucagon prevents hypoglycemia. It actually increases the glucose levels. So what we can see, the system is built up in such a way that you always maintain the same glucose levels regardless of the nutritional state. And the way they do it, that actually they regulate that by different metabolic pathways. And it only makes sense that the glucagon gene will actually include those two, which encode those two peptides. And I think that's a safety build mechanism that you never get to low glucose and which can lead to hypoglycemia and death eventually. So it's a beautifully devised system.
Yeah, I don't quite see it that way. So the interesting thing here is the processing enzymes. That's what determines what comes out of these products. So you're looking at the prohormone convertase 2 in the pancreas that is responsible for the cleavage out of glucagon, and the protein convertase 1,3 in the gut that cleaves out GLP-1 instead and leaves the other parts in an active form. So what is really interesting here is how do we view the biology of these things? If you look at glucagon biology, it's different in different species. So it has developed over the years. It's different in fish. It's different in birds. In birds, it's extremely important for lipolysis. In people, it doesn't work on lipolysis at all. The birds will fall down if they don't have glucagon. So there are some big differences that we don't really understand. And I don't see GLP-1 as the major, as I demonstrated that, the major incretin. GIP in healthy individuals is the major incretin hormone. But what it does is it's located slightly further down the small intestine in the L cells, which are dense there. And when food arrives at the more distal small intestine, where it's not really supposed to be because it's supposed to be absorbed in the proximal gut, right? Then it signals that there is now too much food coming in. And what it does then is signals appetite reduction by vagal actions. It inhibits the stomach emptying. It inhibits the secretion of acid and pancreatic secretion. So it is a signal. It's an enterogastrone. It's an ileal brake hormone that limits, the intake of food. It says this is a signal that now we have an abundance of nutrients there. So that's what it really signals. If by any chance any glucose has arrived in the circulation, okay, then it also stimulates insulin secretion to help get away with that glucose. So that is a neat system. But you're really asking who invented this idea of expressing PC1 in one and PC2 in the other tissue. And that one I cannot answer. You'll have to ask a priest, I think.
But I also want to mention having it as a pro-glucagon, as a pro-hormone in which you have the 37 amino acids, which is inactive. It's also a way to regulate it because you don't want to have 7 to 37 there present all the time. It's probably you need to have it in an inactive form, and it goes back to activation of the enzymes, which will then release it when you take a meal. And so I think that's a very fine mechanism in the body, which also regulates the secretion of 7 to 37. And it goes down to the enzymes, like in some sense.
So yes, based on the homology for the sequence for GLP-1, the truncated form and the glucagon, is that also we call the evolutional conservative?
Yes, that's right. So we know that conservation of amino acids evolutionary are very important in biological activity. And actually, fish has 2 forms of GLP-1. One which is secreted in the pancreas, the other one in the intestine. And actually, I have shown with my collaborators that one form, the one which is closer related to the mammalian, 7 to 37, is more biologically active than the second form, which is less. And the second form has less conservation in the amino acids compared to glucagon. So in fish, there is a different mechanism. So I think GLP-1 and glucagon are 2 very important hormones evolutionary who actually regulate glucose metabolism by different metabolic networks.
Okay, thank you. So we have a discussion of glucagon and the GLP-1. I would assume the sequence difference between glucagon and the GLP-1, there are some sequence
50%,
difference, right? So I would assume those different sequences might have different regulation yet to be discovered in the future. I don't know. I'm a layman, so that's what I thought. But could we also invite Zhang Limin to come in from the question from a physician's point of view? Thank you.
Yeah, I'd like to thank the great discovery of GLP-1 stories. Just heard from the 2 important Tang Prize laureate. It indeed changed our great paradigm shift in management of type 2 diabetes. Previously, we know the anti-diabetic drugs, probably glucose lowering, and nicely to see the GLP-1 to stimulate insulin secretion. However, we now know beyond glycemic control, we also see this class of the drugs might provide organ protection effect. So it's very amazing that we current medicine, we have a very nice class, a new class of the drugs for treatment of diabetes. But my question would be start from the very beginning, that when you know the GLP-1 stimulate the insulin secretion, and also you see later development trying to know the distribution of GLP-1 receptor. But how do you expect such GLP-1 based therapeutics would result in such a huge development in addition to glucose lowering, but also provide so many organ protective effects? Would you like to address some of your comments?
I think, again, it's actually the overall homeostatic state of the organism. And you have to have some coordination of the effects in different tissues. And I think it has to do with that. And we have never looked at the other hormones, whether they have the same kind of effects as GLP-1. This is the first example when we explore the biology of a certain peptide in a different way. I think it's all related. You have to have healthy, kidneys to release, to regulate glucose. You have to have healthy cardiovascular effects, and there are also receptors in the vasculature. So I think it's all has to do in maintaining the whole homeostasis and the whole metabolism in a steady state. And it's remarkable, actually, that was not expected. I must say that at least when we were discovering GLP-1, that it will have so much multitude effects. And we are just understanding that. It's giving us a candle to try and understand that and maybe move on and develop maybe second generation of better peptide-based. That's what I think, Jens.
So we've been very interested in these patients with bariatric surgery. And, they have this huge weight loss, and if they have diabetes, it goes away. So they have up to 30-fold elevations in the secretion of GLP-1, as we've shown in 2005 and several years after. And so initially we thought that the effect on insulin secretion was extremely important for the disappearance of the diabetes. And that's probably also true for the first year or so, and maybe the second year, but then it turns out not to be that important. But what is important is the weight loss. So it's really the weight loss and the improvement of insulin sensitivity that really drives this. So the way I see it, is in terms of its effect on the body composition, the weight and all that, and the insulin sensitivity, that this is really the major effect of the GLP-1s. And you can look back in history to try to understand how this came about. In 1973, CCK was proposed as the first gut hormone to inhibit food intake. So this new concept of inhibition of food intake with a view to treat obesity, of course, was started. So it was already there when we started working with GLP-1. We said right away we would look at this. We would look at these gastrointestinal functions. We would look at the vagal interactions, because this would be the way to do it. We would look at the brain and the food intake. We did all that. And we found that it did all this. So it fits completely in the pattern of GI regulation of body functions and metabolism. And it was just one of the partners that we picked out. And we picked it out because of the insulin release. We went for we wanted to find an insulin-releasing peptide, and we found it.
Well, I think when we were looking for incretins, we knew biology was there. We knew what we were looking for. But I think when it comes to weight loss, it really came from the clinical studies. And yes, we've understood that, but we have to really understand now the pathways, how that's regulated. So I think we have to translate the results from the clinical studies to basic biology, to go back to the bench and understand how the GLP-1 receptors in the brain integrate themselves in different signaling pathways to regulate obesity and other effects. Appetite, which will probably be different than weight loss.
Yes.
Okay.
Sorry. So it's a beauty of integration of basic and clinical science. I think
yes,
we can
that's
demonstrate
right. That's
it,
right, exactly.
Very nicely. Yes.
GLP-1 is an example
really.
of that, in which we can integrate, basic science on the bench with clinical.
I'm just curious about the previous story about the so-called CV safety raised by Steve Nissen in 2007, where when he pointed out some drugs might carry cardiovascular safety. So the FDA actually encouraged or asked all the new diabetes drugs should do CV safety. But if I'm just curious if FDA did not request such kind of requirement for evaluating CV safety or even stretching to CV outcome trials, how do we, or what's your approach to know the cardioprotective or renal protective effect from the clinical studies?
Should I start here? So that's a very interesting story, and it was rosiglitazone that Steve Nissen picked at and accused it of causing increased cardiovascular problems, and it's probably not true, So, which is really a pity for GSK, who developed rosiglitazone, because they had to withdraw the compound, and it was actually quite a good compound. It's a good anti-diabetic, and that's true for all the TZDs. But they, of course, they cause weight increase, and that's not what you can propose these days. But it meant that the FDA demanded all these cardiovascular outcome trials, and I think they've been extremely expensive, and unfortunately, it's the patients who have paid it. I mean, you have to realize this. All these expenses there are put to the prices on the medicine. So, and I know this is a very acute problem, particularly in the United States. so it has its costs. But on the other hand, they have provided us with incredible wealth of information. For instance, the problem with the pancreatitis that was raised for the GLP-1 receptor agonists, first with the exenatide and then with the rest. And it was a really stubborn one. But with the now 70,000 patients that are now included in the meta-analysis, we can rule out this as a major problem for the GLP-1 receptor agonist. And now cancers have come in, as a fantastic thing. And, so there's a recent study showing that in obesity, there's an increased risk of 13 different major cancers. And in the most recent meta-analysis, it was demonstrated that GLP-1 receptor agonist therapy would reduce the frequency of 10 of these 13 cancers very significantly. So it's coming out now, the beneficial effects of it. And it's really very, very interesting that in all these outcome trials also, the number of side effects, serious side effects, is significantly reduced during therapy. This is perhaps not strange when you look at the health benefits that we can obtain with these, but nevertheless, it's very nice to see that the frequency of the side effects is much lower than in the controls.
Yep. Okay, so Taiwan is a democratic country. I have to save 15 minutes for audience, but we still have a few minutes before reaching the 15 minutes. So the panelists, it's a burning question, please.
Yeah, I have a burning question because Dr. Hobbs just mentioned cancer, right? And in your slide, you also emphasized the factor of inflammation. Yeah. And I know a lot of people now are studying for fibrotic disease, for Parkinson's disease. Where do you think the next one will be?
So one of the problems with all the neuroprotection problem is, how can it get to the brain? Can these agents get to the brain? And the ones that we have right now, they don't get to the brain. They enter the circumventricular organs and bind to receptors there in the fenestrae of these capillaries, but they don't enter the brain. So even if we have a lot of receptors in the brain, they are not targets for the GLP-1 from the periphery, but from GLP-1 that is produced in the brain, in the nucleus of the solitary tract. There is a brain GLP-1 system. It's a little bit complicated story. But so all these receptors in the brain are not accessed by the usual compounds. The great excitement will be the small molecules that are under development, because some of these probably most likely will cross the blood-brain barrier and may actually reach these receptors. And if this is important for neurodegenerative diseases, we don't know that yet, but it's likely because it has been studied in in sections, brain sections and brain tissues, you know, and there's very beneficial actions on all of these neurodegenerative diseases, perhaps even Alzheimer's. There will be 2 major clinical trials coming out now, the EVOKE 1 and 2 trials. They're about to be finished this year, I think, maybe next year. So very soon we will know from big clinical trials whether this will work or not. Thank you.
Okay, so do you have a burning question? How about you? Okay.
Talking about the future, we now know GLP-1 can be used in combination with other hormones. And my question is, how many drugs will come soon? And the problem will be the physician side, because we don't know how to choose the best one for each particular patient. So in the era of precision medicine, do you have any idea how to make a physician more easily to understand, to choose combination A, B, or combination A, C, D? Can you?
I think that will be, you know, you have to try them on your patients. I think it's very difficult to predict. We know that some patients, right, don't have to stop taking semaglutide because they have side effects in the intestines. But that could not be predicted. I think it's very individual. And I think as we get more medicines, we will find out ones which will avoid those side effects and will be appropriate for the other group of patients. But I don't think, at least I don't, combination of peptides can help, but it's still a question of trying different combinations, yes.
Okay.
Unless we use artificial intelligence. Maybe that can come into the play, and then maybe they can help us design molecules which can actually fit into them. And actually people are using that, right? The AlphaFold program, that we can see whether we can find other peptides which can fit into the binding pocket.
And you said the outcome would have to be weighed, the outcome from the clinical trial. I mean, that's the endpoint. Yeah.
I think, yeah, we can, yes, we have to use clinical trials. That's where we get a lot of our understanding of how this GLP-1 or other peptide-based therapeutics will work.
Okay, so I have to say thank you to the audience. You guys give me so many questions, and I only have 15 minutes. So let me just quickly go to the question, and those questions, if it's something simple, maybe you can simply answer. For example, okay, I will not mention the name because there are so many people who ask questions. Which one is the most important organ for GLP-1 agonist? Pancreas or brain? Is there any side effect for the brain in the long term? So that's the question. Answer?
Yeah, I can answer that. That's definitely the brain. Because the body's own GLP-1 acts from afferent sensory neurons to regulate centers in the brain that regulate metabolism and all GI functions. That's the main function, and also appetite. So
and earlier you mentioned that some of those drugs has a blood-brain barrier, be able to pass the blood-brain barrier.
Injected drugs mainly act via the leaks in the blood-brain barrier in the circumventricular organs. So it's a different entry rate compared to the endogenous GLP-1 in the body, but it still hits the same kind of nuclei. One from the way of the nervous system, and the other one from these leaks in the circumventricular organs. The area postrema and the median eminence, they're the most important ones.
So the simple answer is brain. Okay?
Yes.
Okay, so the other question, GLP-1 has anti-inflammatory effect. Is there any research that has been conducted related to this? And does GLP-1 has effect on cortisol? Question.
I don't know that.
If we don't know, we just pass. There are so many questions. No?
Is it meant if it has effect on cortisol secretion?
Yeah, does it affect
the other way around would be true, but
cortisol affects the GLP-1, and whether GLP-1 has effect on cortisol.
No.
Let's wait for your research. Okay.
No, I don't think so.
I don't think so.
Okay.
I wouldn't expect that.
So, next question. Since obesity has been classified as A, B, C, D, what is your suggestion for the long-term use of GLP-1 or GLP-1/GIP for the treatment of obesity?
So the ABCD is adiposity-based chronic disease, and it's it's the concept that this is actually a disease, and right in our country it's considered a risk factor. Obesity is considered a risk factor, and I think it's a nice way of having it because it is a risk factor, and so you're prepared to treat the risk, but not necessarily obesity if it's only there, alone. But that concept is changing with this ABCD concept, and we'll have to adjust to these changing views in the world and try to understand. One of the typical questions is, who should receive these agents? And one of my answers could be, why don't you ask the people? Why don't you ask them what they want? I mean, that's a nice democratic way of treating people, is it not? Ask them.
Okay. So if I'm the patient, I have the opinion to say what I want to choose. Okay, thank you.
Wouldn't you feel like that? Wouldn't you agree?
Talk to your doctor, though, and ask what your doctor whether agrees with you.
Okay, so I will try to combine some of the questions who are related. So, okay, there are people ask question. Well, this GLP-1 related compound looks like it's a magical peptide, right? So, can that be used to treat Parkinson or cancer? Cancer, yes.
We actually talked about both of those, didn't we? I mean, there are ongoing trials on Parkinson's disease, and experimentally it looks very, very promising right now. And cancer, yes, all the obesity-related cancers are definitely improved.
So, look like it is a magic pill. Cancer and Parkinson, they all may relate it. Okay. Then on the next question. Is there any possibility where continuing metformin with the dual agonist GIP and the GIP-1R could minimize side effect of CVD or relapse of diabetes? They are thinking about the side effects.
So the metformin, I mean, most of the studies are done in people with diabetes are done in people that are already being treated with metformin. So most of the studies are with a combination, right? And I don't think that we are in a situation where we would recommend people to stop metformin. Some of the GLP-1 agonists are now being tried as a monotherapy, in the beginning. But I think we have to learn a little bit more about it. It seems that there's a nice background that these compounds act on in diabetes, and I wouldn't stand here and propose that you should drop that.
Okay. Okay. So next question, we still have a few minutes. Are there any side effects when used by healthy people, or is it possible to become a healthy supplement and we can use it to prevent diabetes, CVD, and kidney disease, and so on?
I don't think so. I think, again, you have...
I think Wendy is interested in this. Yeah, to try to make money. So, well, apparently this peptide hormone is very powerful and affects many, many diseases. And so this specific question, I think maybe waiting for more research.
Yeah, but we can already say something, So if you look at people, it's already so that about 40 to 50% of people drop their therapy, their GLP-1, after 1 year. And you can ask why that is. Money is obviously a good explanation for that. But there are other explanations also, and one of them is that perhaps one of the most important actions of the GLP-1s is on the brain and on the reward system. So it takes away the reward of eating, of the pleasure of eating, and that may not be fun. So I usually tell the story about the chef we had, who was a brilliant cook, and he loved his own food, so he swelled and swelled and swelled. So he was put on Wegovy, and then he lost weight, and then he couldn't cook. He was uninterested. It was impossible for him to make good food any longer. So he had to stop, and then he stopped and could cook again. Of course, he grew again.
Well, but that's what we think. You have to have adjustment to your lifestyle. Yeah, that's the key.
Okay, there's another question related to lifestyle. Okay, do you think there are any possible way to promote a new lifestyle to patients while having the medicine treatment? I think earlier you said when you take this drug, you exercise.
Yeah, you must do.
Not just drug without exercise, you will be in trouble.
We published a major study in New England Journal of Medicine a year ago or so, where we looked at the combination of exercise or liraglutide 3 milligrams, or each of them separately, and the combination was really, really good. But the most important thing is that the lean body mass lost during the weight loss was minimal if you also exercised. So you came out, as the person asked here, as a healthy individual, in spite of the weight loss, if you also exercised. And I think that is hugely important.
There's an interesting question here. Can this new treatment cure DM2 completely and reverse back to normal? I think everybody like to recover normal. So the question.
The type 2 diabetes, can it reverse it?
So hey, I know the answer to that. So the problem is whether your beta cell capacity is sufficient.
Yes.
That is always the problem. So if you start early with people that have a good beta cell capacity, you can do a lot. So I didn't show you one of these graphs from the SELECT trial, but there is, even in these people that do not have diabetes, there is an increase in A1C level over the 4 years of the study. So people at that age group and with that adiposity, they will have a decreased beta cell function anyway. So this was not affected by the GLP-1 therapy. So there's something with the aging beta cell, and particularly the diabetic beta cell, that we cannot cure with the GLP-1s. So eventually, if you have a person with a very bad beta cell function, you may have to put in addition, some insulin. But that can be done in a very, very elegant way with weekly insulin and weekly GLP-1, so that you can really have a highly efficient therapy of diabetes. It's possible to do that today.
Okay, so next question. Because GLP-1 medication work by lowering blood sugar, concern has been expressed that patients run the risk of their sugar dropping too low. And how to treat it? Good. You worry about it, over-treat it, and then sugar too low. I thought you just eat more sugar. I don't know. I don't know the answer.
But we know that GLP-1 acts only when there is increased sugar levels. So that's the advantage of GLP-1. If the sugar levels are too low, it's inactive.
So there's auto regulation.
Regulation. Yeah. It's not going to act at all.
But unfortunately, there are, of course, exceptions to that rule. And that's exactly how we discovered GLP-1 in the start, because we were looking for people with reactive hypoglycemia after surgical operations on the GI tract, for instance. And that happens because of exaggerated GLP-1 secretion that then stimulates insulin secretion that then causes hypoglycemia. And that's what we call the delayed reaction of hyperinsulinemia, the insulin inactivation time, which is there still as glucose falls, then insulin secretion stops, but there's still a lot of insulin around, and glucose still falls and falls and falls. And you can do that in healthy people very easily with infusions of GLP-1 and glucose. You can produce it. You can produce any level of hypoglycemia. But fortunately, in people with obesity and diabetes, they are insulin intolerant. They're insulin resistant. So you cannot develop that reactive hypoglycemia in these individuals. But in healthy individuals, particularly in people with Roux-en-Y gastric bypass, I mean the bariatric surgery, they do develop hypoglycemia, and that's because of GLP-1 and insulin. And you could do something about it using a GLP-1 antagonist. Avexitide, which is Xentin 9-39, is now being studied for treatment of post-bariatric hypoglycemia.
Okay, I'm trying to be a nice guy, try to complete all the questions, but I just realized this is the mission impossible. But I'll try to get whatever I can get. Okay, for patients with insulin resistance, is it necessary to combine GLP-1 with an insulin-sensitive drug, or my GLP-1 also improve insulin sensitivity? This might be an easy answer or difficult.
No, it's really easy. Yeah. Because the insulin resistance is mainly due to fat. It's mainly due to ectopic fat in the tissues. And once you start losing weight, you will also improve your insulin sensitivity. So it's usually not necessary. So it's not necessarily, metformin, of course, is an insulin sensitizer. So we already discussed that it's usually combined in people with diabetes with metformin. But it's not necessary because it will improve, not by a direct mechanism, but by removing the fat.
Okay. So the next question, I would like to ask if there's a specific age group for the subject of the experiment. You mentioned many clinical trials. That they have any specific age group been discussed?
So I have been along all through the development of these agents, and children is something that we've been very afraid of getting into because it has these trophic actions on various cell types, and you really never know. So we always have to be extremely careful. Now, trials are being carried out and have been carried out also in children. And one of the age groups where we, I think, can expect the best results with the GLP-1, that's in adolescents, in obese adolescents. So they're running a terrible risk of having even worse side effects than elder patients. So, and it works. It's been demonstrated beautifully that it works so well in these individuals. So you can give them hope, of a decent life, because otherwise they are really at a terrible risk of not only the complications, but also psychiatric and other risks, because it's such a terrible situation to be in. So it's really good.
Okay, the science, I'm running out of time, but there are two very interesting questions. Let me just quickly combine them together. So I was wondering if GLP-1 receptor agonists affect gut microbiome. So that's one question. That's interesting. The other one is, does that affect pregnancy when a woman is pregnant? Okay, that's two questions. Yeah.
I don't know the answer to that.
Yes, it does. It does affect the microbiome because you change your diet. And the minute you change your diet, start on the cabbage, as we said, you will also change your microbiome. So there are rather profound changes of the microbiome, both because of the weight loss and because of the change of the diet. Whether there are direct effects of GLP-1 on the diet, I don't know. I'm not so sure. We've actually looked at this in detail, and it's hugely complicated. One of the most complicated papers I've ever worked on and contributed to, I don't understand a word of it. It's from China. It's about the microbiome, and I don't understand a word. So, but the main message, a direct effect of it, no, I don't think so. So the other one was pregnancy. So that's hugely important. Again, it has to do with, is it possible that it could produce, developmental changes? Yes. And that risk is something you always have to consider. Fortunately, a lot of the women who have been treated with the GLP-1 agonists have become pregnant, even if it's not meant, even if it was not advised. And I'm not aware of any malformations in any child so far during the therapy with a GLP-1 agonist. But this is something that you always must bear in your mind, of course.
Okay, I know you're telling me that I'm running out of time, so I would like to take this opportunity to thank the two laureates who give us so many back and forth communication, and in addition, all our panel discussion expert, and thanks for all of them, but also for the audience. There are a lot of questions, very interesting questions, and so I hope this dialogue that today we like to congratulate the two laureates because they are tremendous contribution to the human disease, and apparently from what we just heard, this magic pill probably affected in many diseases. Even we talk about the microbiome is also indirectly affected also on. So, and with this one that I would like to, I think we are supposed to be close this forum, and thanks for everybody's participation. And this is a great opportunity. So before you leave, can I make a suggestion that give a big applause for all of them and for yourself too. Thank you. So, hey, my friend, are you going to announce close or I announce close? You, oh, okay, you are bigger. So before he announces close, so I just remind everybody, weather is nice today in Taichung, but the typhoon is coming. So after the closing, when we leave this auditorium, please be safe. In particular, these few days, typhoon is coming. Back to you, Yang.
Thank you very much, President Han, and our panelists for this fantastic panel discussion. This concludes the forum. We would also like to thank audience for your full and active participation, which makes this forum a great success.